Okay, so we have biosynthesis of fatty acids. Importance, of course, importance of fatty acid synthesis is that when you have extra glucose, you are storing it for future use, and you store it as triacylglycerol in your adipose tissue. Now, the synthesis of fatty acid starts with acetyl-CoA, and they are put together 2 carbons at a time with the use of a very important molecule from your hexose monophosphate shunt, and that is your NADPH. Our body can synthesize a fatty acid starting from acetyl-CoA and additional acetyl-CoAs until there will be 16 carbons. So how many acetyl-CoAs are that? You have 16 carbons. That would be 8 acetyl-CoAs. All right, there will be 8. Now these 8 acetyl-CoAs are used in the formation of what you call your palmitic acid. So our synthetic pathway for fatty acid formation is up to 16 carbons. However, in the cell, you have a system of enzyme. After the 16 carbons are put together to form palmitic acid, you can elongate it by adding 2 carbons, no? And we have what we call an elongation system. Elongates, meaning you can add 2 carbons at a time to make it into the desirable fatty acid that is needed by the body. So it can go to formation of 18 carbons, 20 carbons, 22 carbons, 24 carbons. And 24 carbons or even 22 is quite rare, no? Meaning these are just very, very special. So the usual amount of number of carbon atoms we have, no, that is common, is 18 carbons. Although there are 20, there are also 22, there are also 24. But the common ones, no, the highest number of common ones, highest number of carbons is 18 carbons. Okay? Inhibition of lipogenesis occurs in type 1 diabetes mellitus, and variations in the activity of the process affecting the nature and extent of obesity. So, inhibition of lipogenesis occurs in type 1 diabetes mellitus, or what we call insulin dependent, or also known as juvenile diabetes mellitus. Now, we have what we call here essential fatty acids. Now, when you say essential, it should be present in our diet, essential amount, no, of fatty acids in the diet, because we cannot uh, form these essential fatty acids. Okay. So we will be able to know what they are later on. And these essential fatty acids are converted to eicosanoids. Eicosa meaning 20. Eicosanoids, and these are the following eicosanoids: prostaglandins, thromboxanes, leukotrienes, and lipoxanes. And prostaglandins mediate inflammation, pain, induce sleep, and also regulate blood coagulation and reproduction. Our non-steroidal anti-inflammatory drugs, or known as NSAIDs, NSAIDs, such as aspirin and ibuprofen, act by inhibiting prostaglandin synthesis. Leukotrienes have ability to contract muscles, no? Contractant, and the chemotactic properties are important in allergic reaction and inflammation. Leukotrienes. Okay. So I will be talking about them. So we look at now the main pathway. As we said, acetyl-CoA is our precursor, no? And we require NADPH, ATP, a little amount of manganese, biotin, and bicarbonate as a source of CO2, carbon dioxide. And the product is palmitic acid, the end product. And this is your de novo fatty acid synthesis, only up to palmitic acid. All right. The first step in our synthesis is to convert, or is to form, a compound known as malonyl-CoA. Malonyl-CoA. This is acetyl-CoA with an additional CO2, or additional carbon. And then, of course, with the carboxylation process, there is addition of an acetyl-CoA, so it becomes malonyl-CoA. The enzyme is acetyl-CoA carboxylase. Okay. This enzyme has a major role in regulation of fatty acid synthesis. And acetyl-CoA carboxylase has a requirement for vitamin B complex member, biotin. And this enzyme is also a multi-enzyme complex. Multi-enzyme complex containing biotin carboxylase. So it has many enzymes, as we can show you the figure later. So we have here malonyl-CoA. So we have acetyl-CoA here, and then we have a bicarbonate as a source of carbon dioxide, and then acetyl-CoA carboxylase dependent on biotin. Remember, biotin is a big complex member, vitamin B complex member, and ATP. So this is energy requiring enzyme. Acetyl-CoA carboxylase requires ATP. And you have now your acetyl-CoA. All right, acetyl-CoA. This is a step-by-step reaction, but this is what we need to know. Acetyl-CoA as bicarbonate, acetyl-CoA carboxylase, malonyl-CoA form. This enzyme is dependent on biotin. This enzyme requires ATP. So here is a step-by-step reaction. There are two enzymes, enzyme 1, enzyme 2. You see? So that's why it's called multi-enzyme complex. Okay? Now let's have this. It is a homodimer. Homodimer. So there are two polypeptide chain containing 6 enzyme activities and one particular protein that carries the growing fatty acid, which we call acyl carrier protein. Acyl for the fatty acid, no, as acyl carrier protein, ACP. Okay. So this is now the fatty acid synthesis multi-enzyme complex. This is not the one that we mentioned of your acetyl-CoA carboxylase to form your malonyl-CoA. No, this is now your enzyme. So it is a dimer. One polypeptide chain, another polypeptide chain. This is the ACP that we mentioned, acyl carrier protein. That's the one there, acyl carrier protein. Okay, and you have these enzymes: 1, 2, 3, 4, 5, 6. Okay, 1, 2, 3, 4, 5, 6. Now, if you include this is a carrier protein. It makes it 7. No? So that's one there, and this is another here. Okay. So it is a dimer. So this one is only one. That's a monomer, and this one now is a dimer. Okay. Now, all right, I will transfer this. Someone move. Okay. Can Hindi makuha. Kani mo to siya. All right. Let's look at this one, this chart. Now you have two enzymes. I mean two polypeptide chain. Okay. That is one there and the other is this one. So you see 1, 2. Okay. There are two SH group. There are two SH group. I don't know if this is clear to you. SH, another SH group. No? Okay. Now, the SH group is found in two places. One is with pantothenic acid. So that's it, pan. And another SH group through your cysteine. Diba? Dua. May SH sa cysteine, amino acid cysteine, and you have an SH sa pantothenic acid. This pantothenic acid is there. The cysteine is there also for this particular polypeptide chain, and the other polypeptide chain is also this. So they are inverted. Look at this. Pantothenic acid there. No. The pantothenic acid is in here. The cysteine is here. And the cysteine is here. The other one. Okay. That is the fatty enzyme, fatty acid enzyme, multi-enzyme complex. Okay. The first reaction is this one. Acetyl-CoA converted to malonyl-CoA by your acetyl-CoA carboxylase. So you have now your malonyl-CoA. This malonyl-CoA will be attached to this number 2 here, number 2 polypeptide chain. So there you are. That's malonyl-CoA. So coming from here, malonyl-CoA is attached here in your pantothenic. That one there. All right. Another molecule of acetyl-CoA, okay, is attached to the SH group here. Oh, look, there you are. Did you see that? So you have here 2 carbons, acetyl-CoA, malonyl-CoA, 3 carbons. Okay. There is the that's a high energy bond there, squiggle. Do you see that sign there, squiggle? No? That's high energy bond. So you have 2 carbons here, that is acetyl-CoA, and your malonyl-CoA, 3 carbons, this one. All right. So how many acetyl-CoAs are involved? You have one here. No? Another one here with an additional carbon. So how many carbons will this be? 1, 2, 3, 4, 5. That's important. No? All right. So what happens next? Decarboxylation occurs. Okay? Decarboxylation occurs. As this acetyl-CoA is added to the malonyl-CoA. There you are. No? That carbon is removed as carbon dioxide, and this one is added here. Okay? So you are adding 2 carbons to these 3 carbons here, that would have been 5. But carbon dioxide is removed. So what is left would be 4 carbons. 1, 2, 3, 4. Did you get it? Huh? All right. Next one. So this one now is known as is reacting with this NADPH. 2 hydrogens are added. NADPH, it becomes NADP na lang. And this will be used, this NADP, through your NADPH generators. There are 3 of these generators. The number one, the main generator of NADPH is your pentose phosphate pathway or hexose monophosphate shunt. Then you have your isocitrate dehydrogenase, and then you have your malic enzymes. So these are the 3 that forms NADPH. But the major producer of NADPH is your pentose phosphate pathway. But these 2 can also produce NADPH. But the amount is not so much, it's almost negligible. Even without them, we still can have NADPH courtesy of your pentose phosphate pathway. All right, so that is the first reduction. No? Reduction. You are adding 2 hydrogens there. So look at that. That is oxygen there only. You add a hydrogen, it becomes an OH. You remove the double bond, and the other hydrogen is added to this carbon here. Huh? Huh? Okay. So you have now a hydroxy. Now, still attached to the enzyme. So that's why it's called 3-ketoacyl enzyme. Now it says 3-hydroxyacyl enzyme. So this is the enzyme that is still attached. There you are. Okay. Then remember that that's OH, and that is your hydrogen. Now we are going to remove this OH and this hydrogen here to form water. The enzyme is a dehydratase. And what do we have? It is now unsaturated. So you can see the double bond there. No? 2,3 unsaturated acyl enzyme. Okay? Now, since this is unsaturated, you have a double bond, we need to reduce that by using NADPH again to give the 2 hydrogen there. So you will have now an acyl enzyme. An acyl enzyme. No? So the 2 hydrogens here are given off to that double bond. So it becomes now, okay, saturated. That one is unsaturated. And then it goes back in here. Huh? But carrying also this one. It is carrying this. So how many carbons is it carrying now? 1, 2, 3, 4. Okay? So actually, we are adding only 2 carbons. First, we form malonyl-CoA 3 carbons and another acetyl-CoA, put them together. That will be 5 carbons. No? That will be 5 carbons. However, this decarboxylates, carbon dioxide is formed, so you will have only 4 carbons left. 1, 2, 3, 4. So it goes to be reduced here. It is dehydrated here. It is reduced here again. And then it goes back, carrying now these 4 carbons. Carrying now these 4 carbons. All right. Okay. Now, before this is another malonyl-CoA is added, the 4 carbons here is transferred here. It is transferred here. So you have 4 carbons there. All right. You have malonyl-CoA here. So 4 carbons here. Malonyl-CoA, how many? 7. The next reaction removes 1 carbon. So what we will have here, there will be only 6. You get it? And then it is reduced. You form a hydroxyl. And then it is dehydrated. You create a double bond. No? You create a double bond. Then other reduction from NADPH. Then this becomes now 6 carbons. And now this goes back there again. The 6 carbons will be transferred here at number 1. And then another malonyl-CoA will be attached at this side and then transferred. Oh, this will be transferred and then dehydrated. So from 6, this becomes 8. No? And then it goes that way again, then that goes back again, and then goes back here. So the addition is always 3 carbons in the form of malonyl-CoA, and then there is decarboxylation in this reaction here where carbon dioxide is removed. So it is just as if you are only adding 2 carbons. But you should remember that malonyl-CoA is 3 carbons. But once it is added, no, it becomes, it is decarboxylated. And actually you're adding only 2 carbons. So from 8 goes back again and becomes 10, goes back, another, that's 12, another, that's 14, and when it becomes 16, here, this enzyme will now remove the fatty acid. this fatty acid here, the growing fatty acid, and release it as palmitic acid. And then the enzyme complex goes back empty-handed. Then another cycle now has to go on. Malonyl-CoA, then acetyl-CoA, and then they're together minus 1 carbon dioxide. You have 4 carbons now. Then the same thing happens until the desired number of palmitate now is formed. So that's how it is. You are using a 3 carbon in the form of malonyl-CoA, but malonyl-CoA is acetyl-CoA plus carbon dioxide. Now once it is added there, no, that should be 5. But now remember that when you put it there, it has to undergo decarboxylation. Then you only have 4 left. Did you get it? So the same thing happens every time 3 carbons is added, and then 1 carbon is removed. Then 2 reductive processes, 2 reduction here, another reduction here. Now that's NADPH. Until you have now 16 carbons. So you go around, then go around again until 16 carbons are formed, and then that is released as palmitic acid, and then your enzyme complex becomes empty-handed, and they start all over again. Malonyl-CoA is added here, acetyl-CoA is added here, so that's what it is here. And there, acetyl-CoA there, malonyl-CoA here. Now this one is added here, that will become 5, but it is decarboxylated. 1 carbon is removed, that's why you only have 4 carbons here. Do you get the idea? So it goes that, this way, this goes back again. So you expect that there are 4 carbons here, and then that is transferred there. Remember, it is always transferred there before it has that malonyl-CoA, because that's malonyl-CoA is always attached here at number 2. And then that is fused together, and then remove 1 carbon dioxide. And so you'll have 1 carbon less. And so you're actually adding only 2 carbons at a time. That's why we form even numbered carbon fatty acid, even numbered carbons, no? 2, 4, 6, 8, 10, 12. That is your synthesis of fatty acid. So the steps are here, you can read it. The ingredients are here. And you will notice there are 14 NADPH that are used. 7 carbon dioxides. So which means you have to make 7 turns. So 7 turns. And 7 turns is only 14, di ba? But remember that is loaded with, at first, there are already 4 right away. So there is 16 as a result. But 7 times only, 7 revolutions. Like that. So 7 carbon dioxide is added. 14 NADPH. Why 14? Because 1 NADPH here and another NADPH here for every cycle. So at least there are two, no? There are two NADPH per cycle. And 7 cycles, that makes it 14. That's why you have 14 there. 7 carbon dioxide. How many water are removed? There is water there. You have 6 water are removed. 14 NADP are produced. NADPH expended. And what is left is 14 NADP also. Oxidized. This is reduced. This is oxidized. Okay. So this is the left side of the equation. This is the right side of the equation. There. That's your arrow there. Okay. The primer acetyl-CoA forms carbon 15 and 16 of your palmitic acid. Forms carbon 15 and 16. Because the last carbon that will be added here will have the carboxyl group, COOH. And we know in a fatty acid, the COOH is carbon 1. So the starting point or the primer there will have carbon 15 and 16, the number there. Okay? That is how your synthesis of fatty acid is done. So that's 16 carbons. Palmitic acid. Now what is this? The provision of acetyl-CoA and NADPH for lipogenesis. So where does the acetyl-CoA, where does the NADPH come from? All right. You are familiar with this already. This one. That is your mitochondrion. No? This is only, an illustration. This is not how it looks like. Huh. Okay. You know what is this? There's the name, citric acid cycle. No? All right. You know what is this? This one going down here. This is glycolysis. What is the end product of glycolysis? Pyruvate. Where does pyruvate go? Into your mitochondrion. Okay. And pyruvate is acted upon by pyruvate dehydrogenase complex, forms acetyl-CoA. Now we know acetyl-CoA or any compounds with a CoA cannot pass through membranes. It cannot because of that CoA. All right. But your formation of fatty acid, the palmitate there, is located outside of the mitochondrion. Cytoplasmic. Okay? So how can this acetyl-CoA go out? How can this acetyl-CoA go out? Now remember, in your citric acid cycle, acetyl-CoA and oxaloacetate combine together to form citrate. Now, there is no more CoA because that will be removed, CoA. That citrate will now be able to go out of the mitochondrial membrane through a transporter. Did you see that letter T there? Transporter of the citrate. So it goes out, citrate is here. Now citrate is acted upon by an enzyme known as citrate lyase and breaks that citrate into oxaloacetate and acetyl-CoA. That is how it is here. Oxaloacetate and acetyl-CoA form citrate. Once it is outside, split by this enzyme, in the presence of ATP and coenzyme A, then you will have oxaloacetate again and acetyl-CoA. All right, so there you have how your acetyl-CoA, how is acetyl-CoA provided? No? Acetyl-CoA is formed inside the mitochondrion and it is going out, no, in the form of citrate, so that once this enzyme ATP citrate lyase breaks citrate, coenzyme A is added to acetyl-CoA while oxalate comes in here. Huh? So acetyl-CoA, it goes your acetyl-CoA, malonyl-CoA, and so on and so forth. Then where is our NADPH? NADPH. So here is your NADPH. What is this? PPP. That is phosphate pentose pathway. Pentose phosphate pathway. Hexose monophosphate shunt. That's the source of NADPH. Now here's another one. Isocitrate dehydrogenase. It produces NADPH, which can also be used here. There is malic enzyme, which acts on malate and produces also NADPH. Produces NADPH. And to be used for acetyl-CoA to become palmitic acid. You see now how these are all integrated, glycolytic pathway, citric acid cycle, fatty acid synthesis. Okay, so here now, you will be able to see how glucose can become fat. Huh? How glucose can become fat through here. Now, in our fatty acid degradation or fatty acid oxidation, we form plenty of acetyl-CoA. Now, that happens only during fasting. So during fasting, there is no glucose. So how can we get NADPH? So fatty acid formation during fasting cannot occur. Diba? You want to lose weight, mag-diet ka. Diba? Because once you are fasting, there is no extra glucose that can trigger the activity of your pentose phosphate pathway to produce NADPH. Now remember, for every molecule of palmitic acid, we require 14 molecules of NADPH. You see? The requirement of NADPH, and NADPH is only formed when there is glucose. Oh? Okay? You understand now? That you cannot form fatty acids which will be esterified with glycerol, becomes your triacyl glycerol and stored in your fatty tissue or adipose tissue. And this only occurs when you have glucose. So this occurs during feed state, not in fasting state where there is no glucose. Okay? You understand that? Now, there are only 14 carbon atoms that your fatty acid synthase complex can make. And so, there are requirements in our cells that would require more than 16 carbon atom fatty acids. Maybe 18, maybe 20, no? And so where does this occur? This elongation of fatty acid chain occurs in the endoplasmic reticulum, the pathway, the microsomal system. Elongate saturated and unsaturated fatty acid, no, from carbon 10 upward by again 2 carbons using again malonyl CoA. Now malonyl CoA is 3 carbons, but as soon as malonyl CoA is attached, it is decarboxylated and form carbon dioxide. Okay, and NADPH is again a reductant. And the enzyme involved is the fatty acid elongase, which occurs in the microsomal system, or particularly endoplasmic reticulum. Now, elongation of steryl CoA, this is 18 carbon, in the brain increases rapidly during myelination in order to provide fatty acid with 22 carbons up to 24 carbons for sphingolipids, which is required in the myelin sheath of your nerve fibers. Myelin sheath or myelination. Okay. And the enzyme involved is fatty acid elongase. So to remember this, how do we elongate the fatty acid? So you elongate your elong, no? Your nose elongase. Huh? Generally, Asians are pango. We need to elongate our nose. Elongase. Huh? So you cannot forget that. Oh, this is how it is. So again, it is malonyl-CoA there, and then acetyl-CoA. It goes the same thing. And then there is the NADPH again. And there is the NADPH again. Now, abbreviation for fatty acid is acyl. So that's why acyl-CoA. Instead, if this is palmitic acid, you'll say palmitoyl-CoA. If it is stearic acid, stearoyl-CoA. If it is oleic acid, oleal-CoA. Now, okay, so you have malonyl-CoA, 3 carbons. Then decarboxylated, look at that. Carbon dioxide is removed right away. And then reduction in ADP, another reduction here in ADP. So you can just see how important is your NADP in your synthesis of fatty acid. Whether it is de novo synthesis, when we say de novo synthesis, is only up to 16 carbon atoms. Now, elongation, so ang elongate, so microsomal elongation system for fatty acid. Of course, in the microsomes, in your endoplasmic reticulum. Okay, nutritional states, of course, regulates lipogenesis. Wa kay kaon, walay lipogenesis. Diba? Because we need, look at this, excess carbohydrate is stored as fat. You cannot find here a statement like that when you say fat is stored as fat. No? Oh. It is excess carbohydrate is stored as fat. In many animals in anticipation of periods of caloric deficiency. Just like us, when we have plenty of produce, we have plenty of rice, we have plenty of corn, we have to put it in the granary, no? Storage. Your body can also do that. Your fat is stored in, your carbohydrate is stored as fat in your adipose tissue. Okay. Okay, now. All right. All right, to regulate your synthesis of fatty acid, you need to have this regulated. What is this enzyme? Acetyl-CoA carboxylase. Is an allosteric enzyme is activated by citrate. Okay. Which is, when you have citrate, is increased in concentration in well-fed state and is an indicator of a plentiful supply of acetyl-CoA. Citrate promotes conversion of enzyme from an active dimer to subunits of the enzyme complex to an active polymeric form with a molecular mass of several million. Inactivation is promoted by phosphorylation of the enzyme and by a long-chain acyl-CoA molecule, an example of negative feedback inhibition by the product. Those, if acetyl-CoA accumulates because it is not esterified quickly, acyl-CoA or the fatty acid CoA, because of increased lipolysis or influx of free fatty acid into the tissues, it will automatically reduce the synthesis of new fatty acyl-CoA, which also inhibits your tricarboxylate transporter. Where do you find this tricarboxylate transporter?
Mitochondria.
The mitochondria. This is the transporter for citrate. Huh? And then we have here citrate, which activates. Diba? So that is your tricarboxylate transporter, preventing activation of enzyme by egress of citrate from mitochondria into the cytosol. So wala ni, your citrate cannot go into the cytoplasm. Okay? Oh, there you are. That is how it is. This is the mitochondrion. This is your transporter, tricarboxylate transporter. Citrate from the mitochondrion goes out. There you are in the cytoplasm. And then you have now your fatty acid synthesis. Acetyl-CoA, the first reaction is always acetyl-CoA to become malonyl-CoA. Huh? Malonyl-CoA. Malonyl-CoA reacts with another acetyl-CoA, and then they join together, remove one carbon in the form of carbon dioxide, then you will have 4 carbons. That's the starting. Then that 4 carbons, you will have malonyl-CoA again, and join to the 4 carbons, that makes it 7. But then you remove carbon dioxide, it becomes 6 na lang. So another round, another malonyl-CoA is added, and so your 6 plus 3 becomes 9, minus 1 carbon as carbon dioxide, then you will have 8. Then it goes back again. So it is repeated until you will have 16 carbons. Once you have attained that, then the acyl-CoA is removed. And it is released as acyl-CoA. All right. You know what is the function of this pyruvate dehydrogenase? Where did you meet this? Hello, are you still there?
Pyruvate to acetyl-CoA.
Very good. So this is inside the mitochondrion, and whenever pyruvate comes in the mitochondrion, this is converted to by the pyruvate dehydrogenase complex to become acetyl-CoA, which is fed into the citric acid cycle. But once there are plenty of acetyl-CoA, although it will react with oxaloacid to form citrate, that citrate goes out of the mitochondrion, acted upon by citrate lyase, it becomes again acetyl-CoA and and oxaloacetate. And the acetyl-CoA is now used through your de novo synthesis of fatty acid. So if you have plenty now of acetyl, not acetyl, acyl, meaning fatty acid CoA, this is going to inhibit this enzyme so that there will be no more acetyl CoA that will be formed. Now, when does this happen? This happens when you have plenty of acyl CoA released. Acyl CoA released when your triacyl glycerol is being acted upon by lipases. And the most important lipase there, which starts the breakdown of your triacyl glycerol, is your hormone sensitive lipase. And then your hormone sensitive lipase, you are now flooding with acyl CoA so that once that is formed, no, your acyl CoA, there's plenty of that, then it is going to inhibit this. And those regulating the availability of acetyl CoA, kay this inhibited naman for lipogenesis. No? Inhibiting or regulating the availability of acetyl CoA for lipogenesis. Because you have plenty of this, it inhibits this one. Adaptive enzymes. The fatty acid synthase complex and acetyl CoA carboxylase are. What does acetyl CoA carboxylase do? This acetyl carboxylase. It converts acetyl CoA to what?
Malonyl CoA.
So these enzymes adapt to the body's physiologic needs. So that's why called adaptive. They adapt to the needs of the body. Changes and also through gene expression, which lead to increase in total amount of the enzyme present in the feed state and decrease during intake of high-fat diet or during starvation and diabetes mellitus. When you have a frank case of diabetes mellitus, the patient always becomes slender. Maybe he is obese, but once he has the diabetes mellitus, he is going to become lean. The fat is being utilized because the glucose cannot be used by the cells because of the problem with insulin. The insulin might not be there. No? So, as in diabetes type 1, or the insulin is still there in diabetes type 2, but there is no receptor for insulin. That's the problem with diabetes type 2. You do not have a receptor for insulin, even though you have plenty of insulin. And the insulin cannot do its work. See? Now, feeding fats containing polyunsaturated fatty acids coordinately regulates the inhibition of the expression of the key enzymes of glycolysis and lipogenesis. You see? This mechanism for longer term regulation of lipogenesis takes several days to become fully manifested and augment direct and immediate effect of free fatty acids and hormones such as insulin and glucagon. All right. So I guess you are saturated. We still have plenty of materials to go on. So I think I will just have to stop here and then continue later on. No? Now, my lectures are usually interrupted by lectures of other doctors. The reason for that is these doctors, no, I give way to these doctors because they are practicing doctors. They have some other, what you call this, they are needed somewhere else. And the only time that they are free are around those times that they give us. So we give way to them so they can lecture. Now you will find that sometimes you have another topic, no? Now they have other topics that is not in continuity with ours, but they have to deliver it, no? So it's okay lang. We can just continue whenever they have delivered their lecture. Okay? Now, I do not want you to be fully saturated. I think you have had enough already. So we will just continue next time when we have the time. Now, because next week you will have Dr. Alita to lecture you on you. And then we will have Dr. Barito later on. So we will have to we will just have to continue, no, after them. Okay? So we will just continue our I will just continue our metabolism of these fats because they are pressing, and we want you also to know about the workings of your nucleic acid so that you will be able to understand literatures that are being put out in relation to this virus that is bothering us. So you'll have an idea of how this is. Okay. All right. We'll continue next time.
Thank you, Doc. Yes.
Okay.
Thank you. Thank you, Doc.
Thank you
Thank
po,
you,
Doc.
Doc.
Thank you, Doc.
Thank you, Doc.
Thank you po, Doc.
Thank you, Doc.
Thank you po, Doc.